Okay, so welcome everyone to this time Immune Zoom seminar. Good morning and good evening. So it's really exciting to welcome this time speaker, Dr. Chen Dong from Shanghai Immune Therapy Institute and Tsinghua University. I think I don't have to go through Dr. Chen's CV because I think most of us probably already know him very well. Dr. Chen actually finished his undergraduate in Wuhan University, and then afterwards he started to pursue his PhD in Department of Microbiology in University of Alabama at Birmingham, which after the PhD degree, he started to pursue the postdoc fellow in Yale with Dr. Richard Flywell. From 2000, Dr. Chen started his independent research program in University of Washington School of Medicine, which afterwards he relocated into MD Anderson in Texas until he got tenure and full professorship in Department of Immunology. In 2010, he got promoted as a chair in cancer research program and MD Anderson Cancer Research Center. Afterwards, Dr. Chen made a big decision, switch his career back to China and become a director and vice dean of school medicine at Tsinghua University. 2014, he became a director of Institute of Immunology, Tsinghua University. And then afterwards, from 2016, he becomes Dean of School of Medicine, Tsinghua University. I think a lot of us actually are very familiar with Dr. Chen's work. A lot of us actually get inspired with his work. His work actually mostly focus on the Th17 and T follicular helper T cells, one of the very pioneers in the field, and then also give a lot of insight into these T cells biology and then pathological functions. His lab also study a lot of T cell differentiation and function, cytokine function and signaling, also immune tolerance and tumor immunity. Dr. Chen really got numerous of words. I'm not going to go through all of them. It's going to take a very long time. I think most of announced ones is 2009. He got American Associate of Immunologists BD Bioscience Investigator Award, and then he also got BioLegend William Paul Award in 2019. And I think the most impressive part is that from 2014 to 2019, he consistently get selected as highly cited research. And then based on his achievement, 2019, he was selected as a member of Chinese Academy of Science. And I think from 2020, he's also become a professor in Shanghai Jiao Tong University. Without further ado, I think Dr. Chen's today's topic is regulation of type 17 immunity in health and diseases. So we're all looking forward to your seminar. Please go ahead.
Okay. Thank you, Chuan, for the kind invitation and the introduction. It's my pleasure to introduce some of our work in the recent years. And before that, I was told I should introduce my new affiliation. So after 10 years, almost 10 years working at Tsinghua, so I started to work at Shanghai Jiao Tong University as introduced already by Chuan. One of the missions was to set up the Shanghai Immune Therapy Institute closely with the Renji Hospital affiliated with Shanghai Jiao Tong University School of Medicine. So this institute will focus on research in understanding immune diseases and also finding ways, novel treatments for some diseases. So today I will discuss with you our recent work on regulation of innate and adaptive cells that produce IL-17 in both normal immunity and also in diseases. As trying to introduce in 2005, our lab and the Casey Weaver's lab proposed that in addition to the traditional Th1 and Th2 cells, we have a new lineage of T cell differentiation, and this is called Th17 cells. CD4 T cells producing IL-17, IL-17F and IL-22, therefore representing a new class of helper T cells. So these cells can be differentiated in a different distinct program from those for Th1 or Th2 cells. VJ, Kuchu, Gita Stockinger, and the Casey Weaver showed that TGF beta and IL-6 are very important to initiate Th17 cell differentiation that necessary. And our lab also reported that IL-1 also mediate early Th17 cell differentiation. Following T cell differentiation, we know that STAT3 downstream of IL-6, IL-21, and IL-23 is absolutely necessary for Th17 cell differentiation. And also there are two lineage, Th17 lineage specific transcription factors, our gamma and our alpha, that are induced in response to TCR and cytokine signaling. They especially our gamma T is really sufficient and necessary for Th17 cell differentiation. So in the early days of Th17 discovery, we proposed that these cells conduct different functions compared with Th1 and Th2 cells. We know that Th1, Th2 cells are absolutely important in our immune system against intracellular and extracellular pathogens, respectively. Th17 cells, by producing IL-17, they are also very important for our host defense against infection, because right now we have people who cannot produce Th17 cells or fail in responding to IL-17 cytokine signaling. These patients are very sensitive, susceptible to extracellular pathogens like the fungi and the extracellular bacteria. Is because Th17 cells are very important to in recruitment of immune cells, leukocytes to infected tissues. Also, Th17 cells are not only found in mucosal tissues, they are also very important for mucosal immunity and also maintenance of mucosal barrier function. And certainly, which has drawn most of our attention and also of interest to many people, including drug company, is the relevance of Th17 cells in human diseases, especially autoimmune diseases. We know that blocking Th1 differentiation did not alleviate most of the autoimmune disease models, but in animal models deficient in Th17 cell function, many of the autoimmune disease models are affected, especially the model most of us use these days, which is called experimental autoimmune encephalomyelitis, EAE. And so we also know that targeting IL-17 is is a proven drug therapy to treat many autoimmune diseases, including psoriasis, arthritis, and also ankylosing spondylitis. So, and this highlights the importance of Th17 cells in human diseases. One question that is the key in the field in recent years is how Th17 pathogenic function in autoimmune diseases are regulated. We know that Th17 cells can be found in mucosal tissues, but normally they don't cause any harm over there. They maintain well balance with the microbiota and also with the tissues. But on the other hand, in diseases such as EAE, Th17 cells infiltrate into the target tissue and cause chronic inflammation. So what is the regulation of Th17 cell function in these two distinct contexts? So that this question has interested many people in the field, including our lab. We're intrigued by the findings from Vijay Kutru and David Heffler that thought such environmental factor can influence Th17 cells. Several years ago, Xiaohu Wang, former postdoc in the lab, now a research associate professor at Tsinghua, asked the question whether fever can directly influence T cell differentiation. As you know, fever is induced after infection and has historically been linked with innate immunity and the inflammatory responses to infections. But whether fever has any direct effects on T cells, they were not known. Also, many autoimmune diseases, the patients have been associated with periodic conditions of Th17 cell differentiation. Xiaohu found that the I-17 secretion was almost double under both conditions. So this is highly reproducible and also reproduced by a graduate student in the lab, Lu Li. Lu not only confirmed these studies, but also conducted the RNA-seq analysis. She found that consistent with the FACS staining data, cytokines such as I-17A and I-17F and a bunch of chemokines were all highly enhanced in the expression at 39.5 degrees compared to 37 degrees. And interestingly, if when she took this transcriptome from the 39.5 degree culture, she found that the transcriptome were similar to Th17 cells culture under IL-23 rich conditions, the so-called in vitro pathogenic Th17 differentiation conditions, and also compare with in vivo generated Th17 cells, the Th17 cells induced at 39.5 degrees did not show correlation with Th17 cells harvested from lamina propria, but they were highly similar two Th17 cells infiltrating into CNS in the EAE disease. So therefore, Th17 cells induced at a high temperature are similar to so-called pathogenic Th17 cells generated in vitro and also in vivo. So then we hypothesized that Th17 cells induced at a high temperature may have more potent pro-inflammatory function. So to test this new culture OT2 T cells into Th17 cells at 39 or 39.5 degrees, and before transfer them into recipient mice and the intranasal administration of ova protein to induce acute lung inflammation. In this model, most of the infiltrating cells were neutrophils. So you can appreciate that in the lung tissue, 39.5 degree cultured Th17 cells will induce more neutrophil recruitment into the lung tissue. And also the same case in the bath. So all these indicating that indeed Th17 cells culture at a high temperature are more potent in the pro-inflammatory function. So then we took quite some time to figure out how T cells sense high temperature in producing more cytokines. So Lu read literature and found out protein Sumoylation has been linked with cells cellular response to high temperature, which she confirmed by total Western of cell lysase using anti-Sumo2 antibodies, and also SMART4 downstream of TGA-beta signaling was known to be Sumoylated, and also Lu found that SMART4 was highly Sumoylated following culture at 39.5 degrees. So then Lu tested whether or not Sumoylation was important for Th17 cell differentiation, especially at 39.5 degree. We were fortunate to have UBC9 conditional knockout. We crossed this with tamoxifen Cree to induce selectively induce the deletion in cultured Th17 cells. Lu found that using 37 or 39 degree, first you can appreciate that there was no difference in Th17 cell differentiation under these two temperatures using UBC9 conditional knockout T cells. I must mention, forgot to mention that UBC9 is the only E2 enzyme for Sumoylation. So, so deletion of UBC9 abrogated all the Sumoylation in the cell. Intrinsic at 37 degree, I-17 expression was also reduced in the absence of UBC9. So therefore, UBC9 may be important for T cell differentiation, but not only for 39.5 degree, but also at 37 degree. So then we hope to find a molecule that is that show enhanced Sumoylation following culture at 39.5 degree. That will be our mechanism to mediate Th17 cell differentiation at 39.5 degree. Since Th1, Th2, and the Treg differentiation was not affected at 39.5 degree, we reason that such sensor or reader may not be downstream of TCR or co-stimulation. So then we look at TGA-beta and IL-6 signaling. IL-6 induced STAT3 was not altered by high temperature. But when Lu tested SMART2 and SMART4 knockout T cells, she found that SMART4 was involved in high temperature induced IL-17 expression. Previously, in 2008, our lab reported that SMART4 was not necessary for Th17 cell differentiation culture at 37 degree. Lu reproduced this data using different conditions of T cell differentiation. You don't see much difference when you culture SMART4 knockout T cells. But interestingly, at a 39.5 degrees, SMART4 knockout T cells show reduced but comparable levels of cytokine expression between 39.5 degree and 37 degree, and suggesting that SMART4 was necessary for T cell differentiation, but only for the part enhanced at 39.5 degree. So as I already showed you, SMART4 was Sumolated, and previously the Sumolation sites have been mapped by others in other systems. There are two lysine residues that could be Sumolated. Lu therefore mutated these two lysine residues and then use wild type or SMART4 knockout constructs to reconstitute SMART4 knockout T cells. Lu found that when she used SMART4 wild type construct, she could see restoration of the sensitivity of SMART4 knockout T cells to high temperature culture. So, after reconstitution with wild type SMART4 molecules, could see that high temperature could enhance IL-17 secretion. But when she utilized SMART4 mutant construct that are deficient in Sumoylation, there was no enhancement. So supporting that Sumoylation of SMART4 is necessary for its function to enhance IL-17 production under 39.5 degrees. But what's the Sumoylation doing for SMART4? Lu found that when culture at 37 degree, after stimulation with TGA beta and IL-6, SMART4 was 100% of cells T cells show SMART4 nuclear localization. But when the Sumoylation was blocked, then SMART4 could not be located in the nucleus. So therefore, consistent with the literature, Sumoylation of SMART4 was necessary for its nuclear localization. So that showing SMART4 being critical in at 39.5 degrees in enhancing Th17 cell differentiation and cytokine expression. This allows us to test the function of high temperature in vivo. So we utilize EAE model. So in the EAE model, it has been known that aspirin can reduce the disease severity. So consistent with our data, we found that IL-17A producing T cells were reduced in the EAE mice in the CNS. So then we utilize SMART4 conditional knockout mice using CD4 to mediate the deletion. We found that deleting SMART4 in T cells resulted in amelioration of EAE diseases. And also IL-17A producing T cells were also reduced in the absence of SMART4. And interestingly, when we treated these mice with aspirin, we didn't see further reduction in IL-17A producing T cells in CNS. So therefore, these in vivo data highlighted the importance of high temperature SMART4 axis in enhancing Th17 function and in autoimmune diseases. But how is SMART4 functional at a high temperature? To address this, Lu did ChIP-seq analysis on SMART4 utilizing T cells culture at 39.5 or 37 degrees. And also she selected genes that will be induced in the expression or enhanced in the expression at 39.5 degrees culture in vitro. So using these three data sets, Lu came to 60 genes who are only bound by SMART4 under 39.5 degrees and also show enhancement of expression on the 39.5 degrees. So the genes directly bound by SMART4 and regulated by SMART4 include IL-17A, IL-17F, IL-21, etc. So this was consistent with our previous finding. Interesting, TGF-beta receptor 1 also show enhanced expression. And TGF-beta receptor is the receptor 1 is upstream of SMART4. So suggesting that SMART4 not only upregulated its downstream genes, but also upregulating its upstream receptor to enhance its own signaling. So, so this work suggests that SMART4 is uniquely situated in T cells to sense increased temperature and to control T cell Th17 cytokine expression and also pathogenicity. So these may provide some mechanism, not all the mechanisms for the link between high temperature and autoimmune diseases. So that is Th17 cells. Let me just say again that that not only can secrete IL-17, but also are marked by the expression of our gamma T, CCR6, IL-23, etc. But in our system, in our immune system, there are also innate lymphocytes that are capable of producing IL-17, especially during the innate phase of immune function. These include gamma delta T cells that can produce IL-17, so they also express our gamma T and also the similar receptors, and also ILC3. So these type 3 innate lymphocytes, they produce IL-17, but more importantly, they produce IL-22 in the gut to regulate gut homeostasis and also gut-associated diseases. So these cells also express our gamma T and also the receptors that allow the response to chemokines and cytokines. So let me share with you in the second part our recent story on the regulation of ILC3. It is known that ILC3 come from precursors exist in the bone marrow, and the progenitors migrate into the tissues and mature into cytokine producing innate lymphocytes. But the tissue and the ILC crosstalk and the regulation has not been well studied in the past. So, so our recent work on IL-17D may share some lights. So let me summarize for you the IL-17 family cytokines and receptors. So these cytokines, I already told you about IL-17A and F, which are produced by lymphocytes, but the receptors are highly expressed in epithelial cells in the skin, lung, and gut. So this is a way that lymphocytes may regulate epithelial cell function and propagation. On the other hand, I-17B, C, and E, they are produced by epithelial cells in mucosal tissues. The receptors are expressed by epithelial cells themselves. Therefore, there's autoregulation, but also these cytokines may bind to distinct subsets of lymphocytes. For example, IL-17E, also called IL-25, combine to type 2 lymphocytes, including Th2 and ILC2. On the other hand, ILC3, I-17C is expressed by epithelial cells and bind to type 17 cells, including Th17 cells, and so on. In this cytokine family, I-17D has had been often cytokine with receptors unknown and expression and biological function uncharacterized, which is the topic of my second part. So this part of work was done by a talented postdoc, Jinling Huang. She found that when she looked at the I-17D mRNA expression, the mRNA was enriched in intestine and the lung, as well as spleen. So therefore, she took I-17D knockout mice, treated these mice with DSS for 5 days, and upon withdrawal of DSS, wild-type mice regained their body weight and show repaired tissue structure, but I-17D knockout mice continue to lose weight and show enhanced inflammation in the gut and worsen tissue repair. So then what could be the source of I-17D? Jinling found that before and during DSS, CD45 negative cells were always the leading source for I-17D. And interestingly, there was no difference between SPF and germ-free mice in the expression of I-17D mRNA. So then she performed bone marrow transfer experiments where she found that whenever the recipient mice were knockout mice in DSS colitis, the transfer mice always show reduced colon length and further reduce body weight, and also show aggregated colitis in this model. So these also support that the bone marrow-derived CD45 positive cells were not a major source or functional source of IL-17D in this acute model of colitis. So, then what at that time we didn't know the target or the receptor for I-17D. Jinling therefore examined lymphocyte function in the gut and found that CD4, CD8, or gamma delta cells did not show obvious defects. But on the other hand, Jinling found that there were some IOC defects. First, she didn't see reduction of our gamma positive T cells or innate lymphocytes, or so-called ILC3, and proliferation was not affected. But on the other hand, IL-22 expression was significantly reduced in ILC3 derived from I-17D knockout mice. And it's well known that ILC3 can be subdivided into 3 major subsets, and they all show reduced IL-22 expression. So then Jinling performed RNA-seq analysis using ILC cells from the gut of Y-type and I-17D knockout mice. Indeed, she found that IL-22 expression was significantly reduced in ILC3 from I-17D knockout mice, as well as AHR are important for IL-22 expression by ILC3, and also expression of CD93, a cell surface molecule. And on the other hand, expression of GATA3 and IL-4 was enhanced in I-17D knockout ILC3, suggesting that I-17D is required for the functional maturation of ILC3 and also for inhibition of ILC3 plasticity. So that was DSS model. Jilin also utilized colon cancer and colon infection, intestinal infection models to test the requirement of I-17D regulation in ILC3 function. She found that in the colon cancer, spontaneous colon cancer model induced by LOM and DSS, in the absence of I-17D, colon numbers and sizes were all increased. And also in this model, she observed reduced ILC3 cells, but not Th17 cells. So then in intestinal infection model, Jilin infected wild type or I-17D knockout mice with Citrobacter rodentia, and which require ILC3 for the initial protection. Jilin found that I-17D knockout mice show reduced body weight and also 10 times less efficient bacterial clearance. And also consistent with our previous data, I-17D knockout mice show reduced IL-22 expression by ILC3. So all these models are not only highlighting I-17D function in the gut, but also suggesting a link between I-17D and ILC3 function. So then whether or not I-17D could directly influence ILC3 or regulate ILC3 function. To test this, Jilin prepared I-17D human IgG fusion protein and used this protein to test binding in colon T cells or ILC3 cells. Obviously, you can observe significant binding to ILC3, but not to T cells from the gut. So that suggests I-17D could perhaps regulate ILC3 directly, but we have to identify the receptor. So Jilin searched in the lab and found the macrophage cell line could also be bound by I-17D. So after the binding, she cross-linked and also conducted IP. So in addition to I-17D human IgG homodimers, she could observe a much higher molecular weight protein complex to be cross-linked and immunoprecipitated. So then she cut this band and subject to mass spec analysis, and after which she prepared cDNA for the membrane proteins identify and test binding by I-17A and I-17D. She found that CD93 indeed could be bound by I-17D, but not by I-17A. On the other hand, I-17A could bind to I-17RA, but not CD93. So this suggests CD93 may be a direct binding partner for I-17D. And so Jilin further confirmed the binding in solution using recombinant proteins of I-17D and CD93. So then, so I said CD93 was highly expressed in our RNA-seq analysis by ILC3. So Jilin tested the expression of CD93 in ILC3 cells and find that those cells highly expressing CD93 also show high expression of IL-22. And we isolated this fraction and subject to RNA-seq, and indeed we found these represent functionally matured ILC3 cells. So therefore, CD93 marks functionally mature ILC3 cells. So then Jilin created CD93 knockout mice, and as a result of the knockout, CD93 antibody staining was gone, and also IL-17D binding was gone. So it indicated that CD93 may be the binding receptor for IL-17D. And also in DSS model, CD93 show knockout mice show enhanced body weight loss and associated with impaired expression of IL-22 downstream genes. So then to further confirm that IL-17D regulates ILC3 function through CD93 expression by ILC3, Jilin created a conditional knockout mice for CD93 and cross with RRC, CRE. So in ILC3 cells isolated from these mice, IL-22 expression was reduced in all subsets of ILC3 cells. Also in the DSS colitis model, you can appreciate that these conditional knockout mice show reduced body weight and impaired wound repair or wound healing, and just like IL-17D knockout mice. So therefore, in this study, we identify a new axis of tissue and lymphocyte interaction in the gut. So gut epithelial cells constitutively make IL-17D independent of microbiota. IL-17D combined to CD93 expressed by ILC3 cells and was necessary for the maturation into IL-22 producing functionally mature lymphocytes. And of course, this IL-22 production was important for wound repair, for control of colon cancer and the control of bacterial infection. And I should mention that CD93 does not represent sequence homology with IL-17 receptor family members. So therefore, this is a unique cytokine receptor interaction, and which needs to be further studied for the signaling and functional mechanisms. So lastly, let me thank the students and colleagues associated to the work. Lu Ni and Xiaohu Wang did the first part on fever, regulated Th17 cell differentiation, and Jilin Huang did the IL-17D work. Xiaohong Zhao is our bioinformatic analyst in the lab and helped them to analyze the transcriptome and also ChIP-seq data. Were helped by Xinquan Wang at Tsinghua University in identifying the direct binding of IL-17D with CD93. So lastly, thank you for your time, and if I may answer any questions.
Okay, thank you very much, Chen. I think we can start a question. Shelley, you want to go ahead?
Sure, yeah. This is really beautiful talk, Chen. Especially the temperature enhanced Th17 differentiation. So I'm wondering how much do you think this is evolutionary conserved? Because like, for example, we know bats, like when they're flying, they have actually pretty high temperature, like close to the fever temperature for usually mammals. And the same thing for birds. I think the bird body temperature is close to 40 Celsius. And maybe if asked the question in the other way, which is like, think about reptile or amphibians, which usually they have lower body temperature. So I just wonder, like, if this like mechanism is conserved in evolution.
Yeah, that's a very good question, which I don't have any answer or idea at the moment. I was frequently asked by physicians whether or not we can find such regulation in humans. I have no data either to answering that. So yeah, that's a very good question, but unfortunately I cannot answer.
Okay, cool. And because I think like usually the high body temperature was considered as a like a increase in immune response. And so it can be potentially like from the mechanistic level explained like from two parts. One is like, we know like the chemical reactions will be accelerated, especially like if we increase the temperature, and the reaction rate will be faster. But the other way, I guess, is like through some like these HIF pathway, which like sense the temperature that induce specific like transcription of specific proteins. So how do you think like these two factors play in the enhancement of Th17 differentiation?
Yeah, so our data suggests is transcriptional because SMART4 was involved, and we could see enhanced nuclear localization, increased binding on the chromatin, and also so enhanced RNA expression levels of the cytokines. But these appear to be specific regulation. We don't see globally increase mRNA expression, and we didn't see enhanced cytokine expression by differentiating Th1, Th2 cells. So why that's the case? We're still puzzling. So as you said, this may be involved in host defense. As we know that Th17 cells are important for tissue inflammation in host defense. So perhaps this is a way to enhance inflammation and the leukocyte migration and recruitment in infected tissues. So that could be beneficial to the host. On the other hand, you probably don't want to enhance Th1 or Th2 function. Yeah, so at least globally. So that's my guess. Yeah.
I see. Yeah, I think that totally makes sense. Okay, yeah, thanks so much.
Okay, so Jenny, you want to go ahead and ask question?
Yeah, so really wonderful talk. Thank you, Chen. So I have a question about the different type of IL-17, cytokine subtypes. Are the epithelial cells specialized in some way, or based on the subtype or anatomic location, or? Yeah,
so these grossly, they are all produced by all these cytokines, especially IL-17 B, C, D, E, excluding A and F. They are frequently expressed by epithelial cells in skin and also in the gut. Some are sensitive to microbiota regulation. We found previously I-17B and I-17E, they require microbiota for the proper expression in the by epithelial cells. Rich Loxley and the colleagues pioneer in showing that IL-25 or IL-17E expression or produced by specialized epithelial cells called tough cells, and in the not only in the gut, in the lung, but also in the thymus. Whether or not the I-17, other I-17 family members also show the specialized production by a given type of epithelial subtypes. That is an interesting question that we hope to also deal with. We recently generated I-17D reporter mice, so which may allow us to identify what kind of epithelial cells may produce I-17D and to be able to isolate live epithelial cells ex vivo for characterization.
Great, thank you.
Yeah.
Shu, you want to go ahead?
Hey, Chen, this is really nice talk. I have a couple of questions. One for the beginning part, for the heat shock response. Do you know if this temperature dependence may connect to the Th17-driven disease in different tissues, whether the organs have higher body temperature may be more susceptible to Th17-driven pathogenesis?
Yeah, so that's a very good question. We in the beginning of our investigation, we also wonder about the physiological relevance. So we tested the EAE model, at least in the beginning phase. We found that upon immunization, so first we saw increased body temperature at the immunized site. We often subcutaneously immunize the mice. And then the increase of body temperature was observed in draining lymph nodes, suggesting that during T cell priming, also the local temperature was increased, and then gradually spread out to other parts of the tissue. So we feel that this was meaningful or physiologically relevant process. So during immunization with CFA, which induce high amounts of IL-17 expression by T cells, so this was associated with increased temperature at these draining lymph nodes when T cell Th17 differentiation occurred. Yeah.
Cool. And a follow-up question.
In the body, there are other tissues showing increased temperature that will regulate Th17 cells locally. I think that's a very interesting suggestion, which we haven't thought about in the past. Yeah.
Right, there has been some link of IL-17F with thermogenesis in brown fat or in the adipose tissues. Just wonder if there's connections. Normally, those will be the one that are highly heated during cold challenge.
Yeah, yeah.
And the follow-up question with the heat response really interesting. Do you know if this HSS1 dependent, the simulation?
Yeah, it is HSS1. Yeah, we have attempted knockdown of various heat shock proteins, and they are at least a couple of them are important. HSS1 is one of them. Yeah.
Cool. And there has been some link about simulation with generally the stress response, not just heat shock stress, but also other stresses. And I think Th17 has been linked to high salt concentrations before. Yeah. Do you think maybe the overall IL-17 production or Th17 response generally happens in those extreme stress challenge conditions inside the body as more of a like stress physiology response? Yeah,
so that's a very good point. So Th17 cells obviously have with a very long evolutionary history. So maybe initially they are not only responsive to infections, but also to the need of stress and tissue repair, which also require leukocyte migration and function. So, so I think perhaps in the beginning, these two functions are combined, and also these was innate to Th17 cell regulation that has been conserved throughout evolution.
Cool, thank you.
Yeah, thank you.
Yisong, go ahead.
Hi, Chen.
Hey,
Great talk. Yeah,
thank
so,
you. Thank you.
yeah, so just a couple of quick questions. So one for the febrile response. So you check the simulation. It's like a heat shock response. Do you see change in the polyubiquination? And if you so if you do the profiling and if you delete UBC19, do you see other simulated or polyubiquinated protein gets changed?
We haven't done that. We very quickly address the function of Sumoylation, but we haven't analyzed the relevance of ubiquitination in this regulation. So are you suggesting that by reducing Sumoylation, there may be enhanced ubiquitination that will target certain molecules, proteins for degradation?
May not be. I mean, especially like heat shock protein, right? And heat shock response, there's a lot of polyubiquination that could be heat shock induced, that could be relevant. So that, that could be find something, and could be in some simulated eventually we get polyubiquinated and get, the protein stability or the things like that. and also sort of follow Xu's comments, considering like a tumor situation, right? So there's a spike of fevers locally or something like that. And the other thing quite interesting you found is pathogenic 17 cells is more prone in the higher temperature. I'm just wondering, so in that situation could be relevant. And I think, and the last question I have is, you had a 17D, so I had this effect. So does it work on Treg cells? Have you checked? Or only some ILC3s?
We didn't see binding of I17D or staining of CD93 antibody on CD4 cells. So that includes Treg cells from the gut as well. We also tested in EAE. We didn't find binding to CD4 cells in the CNS either. So in vivo, at least, we can exclude, pretty much exclude the regulation on CD4 cells directly.
Great, thank you. That's great, Chen.
Yeah, thank you, Isa.
Okay, so I have some questions. So one thing is, I noticed that when you put the high degree on the T cell culture, not only switch on the Th17 cells, but also Treg cells get a bit different. Do you think that difference is because of the indirect from the Th17 program difference, or it's directly target on the Treg cells as well?
Yeah, we look at iTreg, we didn't see much difference. But if you put Th17 cell into, if you put T cells into differentiation with TGF beta and IL-6, obviously you get some FOXP3 positive cells. And with increased temperature, that population was reduced. So suggesting the high temperature may influence the balance of Treg and Th17 cell differentiation.
But that's a fact. Do you think it's also directly target on the Treg cells somehow? Or
we don't think so, because there was no effect by the high temperature on iTreg differentiation.
in vitro?
In vitro, yeah.
I see. Okay. So the second question I have is actually related to the IL-17D part, because I noticed that when you induce DSS, although you find that 17D highly expressed in the epithelial cells, but when you have the progress on the DSS, seems like IL-17D increase, and it's irrelevant. It's not related to the microbiome. So is that something because of the tissue damage, which can?
It's not the increase.
It's decrease.
Yeah.
I see. But that's the change. I mean, it's also it's still
Probably
actually,
because epithelial cell damage.
and then you mentioned that IL-17D is consistently expressed on the epithelial cells in the gut.
Yeah.
Is there any situation it will actually be, let's say, promoted, the expression of IL-17D, or it's actually steadily expressed?
It's constitutively produced, and we confirm by antibody study. So it's at a protein level, but right now we only saw reduced expression in the DSS colitis. We didn't see increase of IL-17D expression in other diseases. So that remains a question, whether or not the situations that IL-17D expression may be enhanced, but we just don't know yet. Okay.
My last question is related to the CD93. I'm actually curious about the expression pattern of CD93. Is there any other cells besides ILC3 also express CD93 in the gut?
In the gut, the primary source is ILC3. In other tissues, there are other cells also are capable of expressing CD93, and we're working on those cells.
I see, because I also noticed that you say IL-17D also expressed in the lung, right?
Yeah.
So the knockout mice.
ILC3 does not, lung ILC3 do not highly express CD93.
I see,
So,
okay.
Yeah.
Okay, but these knockout mice have any lung phenotype?
Not constitutively.
Okay. Thank you, Chen.
Okay, thank you.
Is there anyone that have questions? If not, I want to thank again for your wonderful talk, and then really appreciate your the lecture. I think we learned a lot. Hopefully, you have good luck with the recruitment.
Yeah, thank you.
Thank you, everyone. Yeah, good night.
Bye.
Bye-bye.
Bye.